action potential signals calcium channels to open, The calcium then causes the synaptic vesicles to release their acetylcholine through channels on the axon terminal into the synapse. The acetylcholine attaches to the receptors on the motor end plate of the skeletal muscle, which causes the sodium-potassium pumps to open. The sodium, shown in green, flows through the pumps, while potassium, shown in yellow, flows back out. Acetylcholinesterase is in the synapse, which breaks the acetylcholine up into acetic acid and choline, pumping it back into the terminal end of the axon, and is taken back up by the synaptic vesicles to be used again later for another action potential.
The increase in sodium ions on the inside of the cell causes a large depolarization. This initiates an action potential in the sarcolemma. The action potential travels into the sarcolemma by way of the T-tubules and stimulates the release of calcium that is stored in the terminal cisternae, shown in orange. Calcium is also stored in large amounts by the calsequestrin. It aids in releasing calcium into the muscle cell through calcium channels.
Calcium flows over the thin filament and binds to the troponin, pulling the tropomyosin off the active sites, which are on the actin. The head of the myosin is stretched out, meaning it is in high energy state with ADP and phosphate attached. The myosin heads jump to the active sites of the actin. This is then followed by the myosin head changing to the low energy state, releasing ADP and causing the thick and thin filaments to slide over one another, which is the muscle contracting. ATP then breaks the head free from the active site by binding to it. The myosin head is then energized as ATP is converted to ADP.
This pumps the calcium back into the sarcoplasmic reticulum through the T-tubules. The calcium is then stored again in the calsequestrin.
The troponin then covers the active sites again, allowing the muscle to relax.
A whole muscle is made up of fascicles. This is a muscle fascicle composed of myofibers, which contain mitochondria and T-tubules. T-tubules are vaginations into the SR. The smallest part of the fascicle shown is the myofibril, which makes up the myofibers.
The sarcomere is the basic unit of striated muscle. When the muscle contracts, the thin filament slides over the thick filament and the muscle gets shorter. The thick filament is represented in red and the thin in orange. The M line is where the thick filaments join. The A band, which is the dark section of the muscle, is shown here. I bands, which are the lighter section of the muscle, are located here. The Z disc is separated from the thick filament by the thin filament. The H zone is a shorter band found within the dark section of the muscle. This includes the M line in the thick filament.